Let us begin with today's session. The first and foremost is introduction to viruses. Why it is important to study virology. I need not tell you this is a very burning issue right now. We all are sitting at home because of a single tiny virus. So you all know that what a virus can do, right? What havoc it can create in our lives and how it can change our lifestyle and control our living, right? So on the other hand, if we see scientifically why it is important to study, is because most of the times viruses are the ones which cause number of unexplained illnesses, right? We feel fatigue sometimes, we feel feverish, we go to doctor prescribes antibiotics, and then even though after taking antibiotic, if nothing happens, okay, then we say, oh, it could be viral. So what we generally do is everything that is not explained properly scientifically, whatever we don't know, we blame it on the viruses. And that is why we need to study this particular branch. Because of their roles in the disease and genetics, which we have now come to know, it is very important to have a working knowledge of the basic characteristics of viruses. Every now and then we come across viruses and their outbreaks. Can you tell me which virus I have shown on the screen? I mean, I don't expect you to look at the picture and tell me the name of the virus, but at least from the names that are depicted in the picture, can you guess what kind of virus it could be? As you can see, it's an RNA virus, right? It's an RNA virus. You can also see there are spikes on its surface, right? You can see the name of the spikes as hemagglutinin. Influenza type of virus I have shown, and this is nothing but a right now what we are dealing with, a coronavirus, which is also a type of similar category virus. Okay, so RNA virus, if I talk about this, if you see the arrangement, the spikes are very different looking, right? They are overlapping on each other, and there is a protein envelope. They have written, actually it's kind of a mixed protein and a lipid bilayer. But yes, this is a Zika virus. If you remember, very recent times in last decade, we have come across so many virus outbreaks. To recall, 2014 and 2016, between these 3 years, we had an outbreak of Ebola virus. Do you remember how Ebola virus looked like? This is how the Ebola virus looked like, a helical type of structure. Okay, and even Ebola virus had glycoprotein on its surface. Then in 2018, we had a virus called Nipah, which was transmitted from bats to pigs to humans. And then now in 2020, we have coronavirus. So all this is a Nipah virus that is just now shown on the screen. So all these viruses do emerge very often over the years. And because of our lack of information about them, inability to develop vaccine against them, we are in dire need to study these viruses, to study their properties, to understand their mechanism of action and come up with the vaccines to have some control on their transmission, community transmission. So I hope this is clear. Let us move to the brief history of virology. When did it all start? Okay, have you heard of Jenner? Edward Jenner? So Edward Jenner was the one who came up with the vaccine for smallpox, and smallpox was caused by a variola virus. But however, when he came up with the vaccine, he did not know that there is something called as virus that is infecting. He just logically came up with a solution to stop the spread of infection. However, first proper diagnosis was carried out in 1884 by Louis Pasteur when he gave the rabies vaccine, and he derived this term called virus, which in Latin means poison. Okay, 1890, 6 years after that, two scientists, Dmitry Ivanovsky to be very specific, and Bejering. So these two scientists, what they did, there were already bacteria-proof filters already in action. Okay, people were already processing bacteria in media, and they were already studying them in laboratory. So of course we could use different types of filters which you have learned about them in FY. Those porcelain type of filters they would take, and they would do experiments with them, retaining the bacteria on the filter, right? So what these two scientists did, whatever tobacco plant showed a different kind of diseased appearance, they took the extract of that plant and then they filtered that extract through this porcelain filters that were used to retain bacteria. Okay. To their surprise, whatever filtrate they got after filtering that extract, they were expecting that there would be no harmful infectious agent because they had filtered it through porcelain filters that were used for bacteria. But when they rubbed that egg filtrate on a healthy tobacco plant leaf, to their surprise, that plant also got the infection. So they realized that there is something even smaller than the bacteria that is existing, which is able to cause infection in plants. And that is how they came up with the first virus, that is tobacco mosaic virus. Okay. Although Louis Pasteur gave a vaccine to rabies, he was unable to say that it is caused by a virus. Okay, he just said that there is something which is causing infection, and he derived the word virus because it was poisonous or infectious for that particular uh, rabies infection. Then in 1898, another two scientists worked together, Frederick Loeffler and Paul Frost, who came up with the disease in cattle, which is called as a foot and mouth disease. Okay, FMD, very well known, and a second in line after TMV to get discovered, which was a cause of a viral infection. And then it followed a number of experiments over the decade, which then revealed lot of information about physical properties of viruses, chemical properties of viruses, and their biological properties. Okay, this decade was the one for these kinds of experiment which revealed the size, shape, structure, and all the details of the viruses. And then came 1915 when Todd and Herrle, Frederick Todd and Felix Herrle, they discovered bacteriophages. So this totally turned all the fields upside down because till then what we understood was viruses were able to infect only plants and animals. But now with bacteriophages, everybody understood, oh, they are not the only ones, they are infecting even bacterial cells. Okay, so this gave a new idea to different kinds of research proposals, because nowadays we are also thinking of using phage therapy, right? So this was the whole idea of discovery of bacteriophages for a betterment. And then by 1950s, now what we have understood is viruses play a very important role in cause of cancer as well. Okay, cancer is one of the leading causes of deaths throughout the world, different countries in the world. So viral infections being one of them, so almost 50% of the, not just viral infection, but of course lifestyle and other exposure to oncogenic agents also matter a lot. But yes, we have seen that many viruses are able to cause cancer like conditions because they are genetic modifiers, like they infect our cells and they modify the genetic information, because of which they are able to cause cancer. So this was a brief history of say around 75 years of extensive research that happened when we got introduced to the new field of virology, and today you see how important it has become. Position of viruses in the biological spectrum. That's a very critical kind of topic, because what happened, people realize that this is not something that we have studied earlier. This doesn't look like a cell. It doesn't have nucleus, it doesn't have cytoplasm. Then what is it? I mean, how to define it? It is not even a cell. So should we call it a biological entity? It is not living. It is living only inside the cell. So should we really call it and study it under biology? That was a first question. But then yes, there was another group who said yes, why not? Because it is causing infection, right? So it is ultimately going inside a cell. So we are supposed to call it as a biological entity only. And the next question was, are they microorganisms? Because microorganisms also do have some kind of specific cell structure, isn't it? So there was a lot of debate on this issue, whether to include them in the microbiology or not. And people finally decided that yes, it is very you know very difficult to define it that way, but yes, we can definitely call them as an infectious particle, if not cell or organism. Okay, so we will call viruses as infectious particle, and we won't call them a cell or an organism. And we will also not call them alive or dead, because they are on the threshold, as we know. We can call them active or inactive, right? So even in COVID, we see that they remain active in the air for almost 4 to 8 hours, right? So they are not alive, but they remain active. Active meaning they are able to cause infection. So we will be very specifically saying this while talking, that viruses are not cells, they are not microorganisms, they are infectious particles, and we say they are active or inactive. We don't call it as live or dead. Okay, let us move ahead and let us see properties of viruses. Now, as I said, this is the first reference book that has been introduced to you in this particular year. So we have Terry Shores by Viruses. Unfortunately, there is no e-book or e-copy available of this book. So whatever I'll be dealing with is what information I had read from this book, plus some of the properties that are very common. Okay, so this chart that I have placed on the screen is from Valero. Of course, the properties of the viruses won't change, so there is no harm in reading from another book. But as I said, if I come across new information, anything extra that Terry Shores gives, then I will add up later. So to revise whatever you have learnt, they are obligate intracellular parasites, right? I think FY you had learnt about all these properties. They are obligate. Obligate meaning compulsory. They will have to live inside, and they are parasites, meaning they don't do anything on their own. Everything they snatch from others, and they are seen to infect all types of cells. So bacteria, protozoa, fungi, algae, plants, animals, all types of cells they can infect. Size, if you ask me, they are very microscopic. So we have to use electron microscope to visualize them. But in few next slides, we will understand how vast variety of sizes they exhibit. Okay, so starting from 20 nanometers to up to 450 nanometers, and even larger than that. Some of the viruses are even larger than bacteria. Okay, so such viruses are also known. They are not cells, as we just discussed. They are just the infectious particles. Okay, and they're very simple, compact, and economical. Economical because they don't have to use any energy, right? They do everything inside the cell and use cell's energy. So very simple and compact. Do not independently fulfill the characteristics of life. Of course, they are not living. That's why we can't call them alive when not inside the cell. They are inactive macromolecules outside the host cell, active only inside the host cell. They are geometric and can form crystal-like masses. Of course, we'll see this point more in detail when we talk about the structure of the viruses. Basic structure consists of protein shell, that is capsid, surrounding nucleic acid core. Nucleic acid can be DNA or RNA, but not both. That's a very important point, but not both. Okay. Nucleic acid can be double-stranded DNA, single-stranded DNA, double-stranded RNA, single-stranded RNA. And there is one more point in single-stranded. When we will discuss about nucleic acid, I will ask you that. But this is very unique. In general cells, we have seen our DNAs as double-stranded DNA and RNAs as single-stranded RNA. But viruses, it can be single-stranded DNA and double-stranded RNA as well. Okay, molecules on viral surface impart high specificity and attachment to the host cell. The very important point, because without those molecules, viruses will not be able to attach or adhere to any kind of cell. Okay, and if adherence is missing, then virus won't be able to infect. So basic point of target of development of vaccine is these molecules on the surface of the virus, which will not be able to adhere to the cells, other cells. Then they multiply, taking control of cells' genetic material and regulating the synthesis and assembly of new viruses. And they lack enzymes for most of the metabolic processes. So they will have genes required for the synthesis of their own proteins. Okay, these were the properties in brief. Now let us talk one by one in detail about the general structure of viruses. Now I assume that you have revised your F5 syllabus. Coming back to Toledo from this section, size is ultra microscopic and requires electron microscope, as I told you. It is warped by host cells, meaning inside the host cell it multiplies. So small viral progeny viruses can combine and they burst out in from the cells. And yeah, if we talk about the viruses size, shapes, okay, there is a cylindrical kind of a virus, geometric like icosahedron symmetrical virus, or you have just a circular looking virus. So it's very difficult to understand such viruses in particular, mold it in the particular type of size. But then if we talk about cylindrical viruses, their length is 800 nanometer. Okay, whereas if you talk about their diameter of the cylindrical viruses, it comprises only up to 15 nanometers. So that way, lengthwise it is very big, but the diameter wise it is very small. While pox virus, if you consider it, is 450 nanometer, while that of parvovirus is 20 nanometer. Earlier, foot and mouth virus, the one that was discovered for the infection, reason of infection to cattle, that virus was 21 nanometer and known to be the smallest one. But this one, parvovirus, is yet 20 nanometer. I'm sorry for the spell mistake here. Okay, about 20 nanometer. Okay. So coming to the new type of virus, I am not sure if you have heard of this. This is called as a Mimi virus. Okay, Mimi virus, and now something called as Mama virus is also known. So what is this Mimi virus? Okay, this was first isolated in 1992 when scientists were looking for a cause of pneumonia outbreak. Okay, in UK, Bradford, UK. So what they did was they isolated some kind of entity, and they named it, they thought it's a bacterium. Okay, so they named it as Bradfordococcus or Bradford coccus, just how we have Staphylococcus, Bradford coccus, and they maintained it in refrigerator. Okay, they wanted to study it later, so they maintained it. Later, a decade later, there were another scientists who came up with the Bradford coccus, and they were studying it. To their surprise, they wanted to isolate their cell wall. Okay, so but they could not get any cell wall as such. So they studied it more in detail, and to their surprise, what they found was it was nothing but a virus. And they found under electron microscope, when they observed the structure, they could see icosahedral virus particles. This virus was able to infect insects, fish, and frogs, meaning in animals. Later, the same type of virus called Mama virus was reported in 2008. So how this virus look like? So this is a structure, very peculiar structure, and you can see how big it is, right? Unlike the other viruses, this is a quite large kind of virus. You can see there are surface fibers here. They have written. They have not mentioned that it is a peploma or spikes or something of that sort that we have in general. So it is a non-enveloped virus, right? It has got a protein capsid, and it has got a DNA or RNA, whatever nucleic acid. So it has got a nucleocapsid, but it doesn't have a envelope. To this nucleocapsid, it is showing presence of fibers, right? So it's a very different kind of structure, and this was observed in amoeba, okay, in the lake water. So then came a new term as scientists tried to study this further. They understood that there is something called as virophage. Okay, just how we have bacteriophage, we have something called as virophage. So what is a virophage? Okay, so virophage is something wherein a virus requires another virus for causing infection, something called as a co-infection. Okay. Earlier it was known that no two viruses can infect the same cell, right? But then that's not the case. Virophage is something, this one virus cannot be able to infect the cell unless another virus has already infected, or another virus infects and then it goes inside, and then along with uska machinery use karke it can reproduce. So virophage is a very new kind of concept which we have not much studied, and much of a study is yet to be done in that section. But there is something called as Sputnik virophage. Sputnik. Okay. This Sputnik virophage, why we are talking about, is because it requires this Mimi virus particle and for co-infection. Okay, so viruses like Sputnik that depends on the co-infection of the host cell by helper viruses are known as satellite virus. So this Sputnik virus is called as a satellite virus because it requires co-infection, especially of mama or mimi viruses. And if you search more on the internet, you will be able to get a lot of information about how the cell factories of mama virus is utilized by this Sputnik virophage for reproducing its own type of viruses. Okay, so I think this slide was very new to you, and I hope you have understood all the concepts of this. Now coming to this general structure of viruses, about observing it under microscope. As I told you, we require to have electron microscopy only to observe this kind of structures. But then we have two different methods. If you just have to observe the superficial, like its outline of the virus, then we may use a negative staining, as we do it in the bacterial cases, I mean bacterial stains as well. But if you want to observe the internal structures, then we have to use shadow casting or a positive staining for internal details. So we will learn about this when we talk about the analysis or assays of viruses, okay, more in detail. So right now, you just remember that there are two ways that we can visualize the viruses under electron microscope. One is called as negative staining and other is called as positive staining. There is something called as shadow casting as well. Okay. Now, when we come back to the constituents of viruses, we have something called as the cover of the virus. It could be either capsid, as we just now saw, some viruses don't have envelope, or the viruses could be enveloped. In that case, the cover would be of envelope. So can anyone tell me, if you have revised about your FY syllabus, what is the capsid and what is the envelope?
Capsid is the protein covering.
Capsid is the protein covering, correct. And what is envelope?
Membrane made up of lipids.
Okay. Is it that envelope is only made up of lipid?
It's also made up of proteins.
Yes. So is it that the proteins of the envelope are same as that of the cells? The question is it that the proteins of the envelope of the virus are same as that of the proteins of the cell membrane?
Yes, ma'am, it is same.
Okay. It is same. Zubi says it is same. How many of you are agreeing? Rachi says no. Okay. What else do you know about viruses? Other than capsid, what else is there? Come on, tell me what you have revised. So then we will proceed with the PowerPoint. No, Prachi, you are saying that no, meaning you are saying the proteins are same or different? Different. So, but then you are saying that envelope is something that virus attained from host. Then how can the proteins of the envelope be different than the ones that they have attained from the host? I mean, correct me if I'm wrong. If you are saying that virus attains envelope from the host, don't you think the proteins of the envelope will also be the same as that of the host? Anything else you remember about viral structures? We are just up to capsid and envelope. What else do you know about viruses? Spikes for attachment of host. Attachment with host. Correct. Very good. So are these spikes present always only on envelope? And what is the property of spikes? What are they made up of? Have you all revised? Yes, very correct. They are glycoproteins. Glycoprotein meaning they are carbohydrates and proteins in nature mixed. Okay, can you give me an example of such glycoprotein? Yes, GP127 on what? Which virus? No. Yeah, but yes, 120 and GP41. Okay, 127 nahi, 120 hai. HIV, GP120 and GP41. Okay, we will see the structures in due time. Let us go back to our presentation. Yeah. So viral constituents cover that will have capsid and envelope, or it can be naked virus. If we talk about the core, it could be nucleic acid and enzymes. Nucleocapsid is the one which is combining of both capsid and nucleic acid. Okay, so nucleocapsid. No protein synthesizing machinery is carried by the virus. It is using cells machinery. Okay, this is a very important point that I wanted to discuss with all of you. What is capsomer? Capsomer, as you would call it, as subunit of capsid. But then have you heard of something called as protomer?
Yes, ma'am.
So what is the difference between capsomer and protomer? That we will see now. Arrangement of capsomers decide the symmetry of the virus. Okay. So coming to the point which we talked about, protomers. So protomers are nothing but the group of proteins. So if you talk about these proteins as shown in the diagram, these are the small proteins coming together and one unit is formed. Okay, so this two pink and one yellow is called as one protomer. So like this 5 protomers come together and form capsomer. Okay, so capsomer, although we call it as a unit of capsid, actually capsomer is not undividable, as we call it as a unit. Protomer is divided into small protomers, right? And protomers are made up of proteins. So here you can see 5 different protomers have formed one capsomer. Like this, different viruses will have different structures. There could be 5 protomers, there could be 3, there could be 6 protomers. Okay, it all depends upon virus to virus. This is a very important difference that everybody should know, the difference between protomers and capsomers. Okay? Protomers are monomers that make capsomers, and protomers self-assemble to form a capsomer. Whereas capsomers are the basic morphological subunit. This is a very important term, morphological. They also self-assembled to form capsid of the virus. I hope this concept is clear. Capsomer and protomer. If not, at the end we will revise. Coming to the symmetry after the formation of capsid. The symmetry is again in 4 different parts, we can say, or 4 different types we can see viral symmetries, wherein if the capsomers bind to each other to form a hollow disc, okay, then it could be one above other and place it like a cylinder, then it becomes a helical type of virus. Just now I showed you the diagram of Ebola virus. So that could be a helical symmetry. But a icosahedral symmetry is a 3D structure like a diamond or something, how it will look like. But then it's a hexagonal 3D. Okay. So it has got 20 sides, 12 vertices, so on. So this part we just talked about. Protomer is the one that forms capsomer. And there are two other types of symmetries. One is binary and fourth one is complex. So binary is the one which is made up of like half of the part of the virus would be helical, half of the would be icosahedral. But complex is very complex, like we are not able to define it what kind of symmetry it is. So one of the very prominent and peculiar example is a pox virus. Here you can see it on the diagram. That is parapox virus. And you will say that it looks similar to what we saw in the other viruses, but then there are foldings. Okay, as you can see here, they have mentioned as surface filaments, right? So these filaments are, we are not able to define what kind of structure they show on the surface. So that is why this is a very complex type of symmetry we call for this particular type of virus. Then we have viral envelope. We talked about Proteomers, which is nothing but the spikes of the viruses that protrude from the envelopes outside, and they are also very important for the adherence. They are made up of glycoproteins, and generally enveloped viruses bud off from the cell membrane. So when they go, they adhere, they leave their envelopes on the surface, and when they are budding off from the cell, from the same particular point, they will bud off, taking out their covers. Okay, they could bud off from the nuclear envelope, they could bud off from the endoplasmic reticulum as well. Now this was the point, as you all said, that the proteins of the envelope and proteins of the host cell membranes are same. But do remember this, that the proteins of the viral envelope and proteins of the cell membrane are not the same. Virus replaces too many proteins of the cell membrane after the budding off, so that it could replace it with its own proteins, viral proteins. Okay, this is a very important point. As far as our understanding is concerned, we always remember that virus takes off the cover of the cell, right? So it should be the same, but it is not. So viral proteins replace the cell proteins. I hope this is clear. If we talk about the function of the envelope, it holds capsid, function of the envelope proteins, to be very specific. ये viral proteins क्यों replace करने हैं? Because these viral proteins function to hold capsid and envelope together. Okay, or sometimes it can also hold the surface receptor, which we talked as glycoproteins, right? So these are very important proteins, and this point should be remembered. Envelope is supple as in soft. Okay, envelope is soft than capsid, and it tends to make the virus pleomorphic. Okay, when I say pleomorphism, okay, that means a single cell shows different forms throughout the life. So a single virus, because sometimes it has got envelope very soft, may show different kind of structures. Are you all understanding this? This is what we call it as a pleomorphism. Have you all understood this?
Yes, ma'am.
Okay. These are some examples just for showing you what is Proteomers. Okay. So glycoprotein type 1 and glycoprotein type 3 on the cauliflower mosaic virus. This is the HIV structure that we just talked about. Glycoprotein 120, 41, okay, embedded in the membrane. All this we will be studying in detail later. And that's, I think, the last slide of the presentation. Viral nucleic acid, as we discussed, either DNA or RNA, but never both. That could be single stranded or double stranded. Now comes an important point. This I wanted to ask you. Do you know what is sense, positive sense and negative sense in case of single stranded RNA?
No, ma'am.
Okay. Simple word should I define positive sense? The viruses which has single stranded RNA, and that RNA can undergo translation in the cellular machinery. That will be called as a positive sense. The viruses which have negative sense RNA will be required to follow one step more. कौन सा रहेगा वो step? Getting a complementary strand. I hope this is clear. And what we are left with is the genes. How many genes a virus will have? Not more than what are required for replicating its genetic material and giving out the capsid proteins. Okay, so genetically also it is very light. We have very less number of genes in viruses, and what we are left with the examples of enzymes. So give me any one or two examples of enzymes and their functions.
Ma'am, reverse transcriptase.
Correct. That's a very good example. Reverse transcriptase possessed by HIV, which is used
Yes.
to convert RNA back into DNA. Like that, we have integrase, we have polymerase, sometimes RNA dependent RNA polymerase. Many different types of enzymes are there. We will learn about them one by one when we talk about different types of viruses. Okay. I hope the lecture was audible enough and you have understood all the concept. If you have not, please let me know before we begin and continue with the other concepts next session.